Bismillahirrahmanirrahim. Today we will discuss about the mechanism of enzyme action, that is the topic in chapter 7, bioenergetics. Before discussing the mechanism of enzyme action, you must be familiar with some basic terms like what is enzyme. Enzymes are the proteins that act as biological catalysts. They can alter or speed up chemical reactions. As they are proteins, so they have three-dimensional shape. They have specific structure and they act as biological catalyst, which means that they only work in living organisms and they change or they speed up all the chemical reactions that are taking place in living organisms. What is active site? Active site is the site where the substrate binds with the enzyme. It is the region on the enzyme which is taking part in the chemical reaction. And what is substrate? Substrate is the substance on which an enzyme acts. And what are the products? The substances that are formed after chemical reaction. Next is the mechanism of enzyme action. First of all, enzyme which has specific active site, it will bind with the specific substrate. When the enzyme bind with the substrate, it will form enzyme substrate complex. After some time, enzyme substrate complex will dissolve into products, and at the end of the reaction, enzyme remain unchanged, and now the enzyme is ready to react with another substrate. Look at this diagram. First of all, this is the active site, which is the site that is present at the enzyme and that is involved in the chemical reaction. And this is the substrate that has specific shape and that will bind with the active site of enzyme. When this substrate will bind into the active site of enzyme, it will form enzyme substrate complex. When the enzyme will bind with the substrate and form enzyme substrate complex, chemical reaction occurs and enzyme will attach to the substrate. After some time, enzyme substrate complex break into products, and at the end of the reaction, enzyme will be set free, and now this enzyme is ready to react with another substrate. The mechanism of enzyme action can be explained by two models. First is the lock and key model. This model is proposed by Emil Fischer in 1894. According to this model, active site is a rigid structure. As active site is a rigid structure, it means that the active site cannot change its shape according to the substrate. So only the specific substrate binds with the enzyme. So in this model, enzymes act as a lock. So only the enzymes bind with the specific substrate and substrate act as a key. Look at this diagram. According to lock and key model, active site is a rigid structure. So it cannot change its shape according to that substrate. So these substrate which have specific shape that fit into this rigid active site is only bind to that enzyme. When the substrate will bind into the active site of enzyme, it will form enzyme substrate complex. When the enzyme substrate complex is formed, so the chemical reaction will start. When the chemical reaction will start, the enzyme substrate complex will be changed into enzyme product complex. This is enzyme and this is product. So enzyme product complex is formed, which is dissolved into products, and at the end of the reaction the enzyme remain unchanged, and now this enzyme is ready to catalyze a new reaction and to bind with the new substrate. Next is induced fit model. This model is proposed by Daniel Koshland in 1958. According to this model, active site is a flexible structure, so it can mold itself according to the substrate. As the active site can change its shape according to the coming substrate, so that this model is more acceptable. Like, look at this diagram. According to this model, active site is a flexible structure, so it can change its shape according to the substrate. So now, no matter which substrate is going to be attached with the active site, the active site has the ability to change its shape according to that substrate. When the active site binds the substrate, it will form enzyme substrate complex. When the chemical reaction started, that enzyme substrate complex will be changed into enzyme product complex, and after some time the enzyme product complex will be break into products and the enzyme, and now this enzyme is ready to catalyze a new reaction. Thank you.